Hi, hi, hi. Okay, it's working now. Well, first of all, I'm very happy to be here in Alive presenting my work on the synthetic microbial Daisyworld. I'm based in Barcelona in the Complex Systems Lab. And I hope at the end of the talk you will understand what this picture is about. Without further delay, let me guide you through a paradox at the planetary scale. For example, if you want to calculate the temperature at the surface of the Earth, it's pretty easy. You just need few physical assumptions and the Stefan Boltzmann law, which correlates the temperature of a body and the thermal radiation it emits, which correlates again with the energy that, in this case, comes from the sun, right? If you do that, you will end up in an expression like the one you have on the top of the slide, that gives you a result of minus 18 degrees, right? And this is not the Earth where we live at, right? This would be a frozen planet. There's a correction that needs to be made on this calculus, and this correction is about the greenhouse effect. To make this correction, you just need to add this epsilon at the denominator in this expression at the bottom of the slide. And this gives you a result of a chilly but comfortable temperature of 15 degrees, the planet we live on. But when you think in geological timescales, then it comes a paradox. If you think 2.8 billion years ago, when life started, we know that the sun was 70% dimmer. So if we do the calculus again, we end up again in a frozen planet, no, in a snowball Earth. And then here it comes a paradox. How can it be that in such a planet with no liquid water, life could have started? Then Carl Sagan and George Mullen came out with an hypothesis. Now there is consensus about it that back then there was a much stronger greenhouse effect. But then it comes a second paradox. If the greenhouse effect was bigger, was higher, and the luminosity has been increasing, then why we are not living in a hotter Earth, right? Why we are not living in a planet like Venus, where the greenhouse effect is totally uncontrolled, or even in Mars, where there is a complete lack of it, right? Instead of that, we know that the Earth has been like traveling through time in an homeostasis that has been allowing liquid water to exist and life, right? And it has been decreasing the greenhouse effect just at the right amount. So what is the mechanism behind that, no? Behind this homeostatic state that Earth is in? Well, James Lovelock and Lynn Margulis came out with a very beautiful and interesting idea, and it's about that. No? It's like the biosphere itself has been the responsible to modulate the atmosphere in its own benefit, in a totally self-organized manner, regulating its own environment to an homeostasis. Actually, you can find examples of that of this planetary physiology out there. I just give you two of them. The first one is the production of clouds from phytoplankton blooms. These clouds, being white, they have a high albedo, therefore they reflect solar beams and they cool down the planet. An opposite example would be boreal forests, right? Where with low albedo, they absorb luminosity and they hit themselves and they hit the planet. James Lovelock and colleagues, in order to provide from a theoretical framework to this idea, they came out with a very classical model that is called the Daisyworld, that probably many of you know about, but if you don't, it's a very simple idea. You just need to think about the planet and its star. This star, like our sun, is increasing in luminosity, and the planet only contains two types of species, black daisies and white daisies, right? They share the same metabolism, so they have the same optimal temperature to grow, 25 degrees, but with one difference. Black daisies absorb luminosity because they have lower albedo, and therefore they heat themselves and the planet. And black daisies, they do the contrary. And if you look at the dynamics of this system, these graphs, you can see in the x-axis the solar luminosity. When the solar luminosity is low, meaning that the planet is cold, the black daisies can grow, no? They can heat themselves and heat the planet a bit, rising the temperature. And when the temperature is too high because of black daisies and the solar luminosity increasing, then there is a shift, and then white daisies outcompete. These end up in these population changes that allows this homeostatic range to exist. So they poise the temperature of the planet just at the optimal temperature for these two species in a totally self-organized manner, right? This model has been very useful to understand the great planetary transitions, right? But like Tim Lenton and Michael Mann, they talk about it in these two great books. But there is an experimental framework to test this theory is still missing, right? And this is, since this model is only theoretical. And this is what we were thinking in Santa Fe 1 year ago. And we came with the idea of how to actually test this model, test this hypothesis in the test tube, in the lab. So if you think about it, all the ingredients were there, right? Instead of a planet, think about a bioreactor. Instead of white and black daisies, think about genetically modified bacteria. Instead of temperature and luminosity, think about pH. So in a schematic way, think about L is luminosity. Instead of luminosity, you have the income of pH that comes into the tank. Like you can introduce acid or base into the tank externally. Instead of temperature of the planet, think about the pH inside the tank. Here, the pH free. Instead of white and black daisies, think about acid producers and base producers. To do that, each strain would contain a genetic circuit that we designed that will constantly produce acid in one strain and base in the other. To check that this approach, this experimental approach, could have the same dynamics, the same mechanistics that the Daisyworld, that the classical Daisyworld, we developed the model for it. For that, we came out with two differential equations, one equation coupled between them and in competition, and one of them for the acid producer and the other for the base producer. The important thing here to look is beta. Beta is the growth rate of this of each strain, right? This beta, this growth rate, follows an inverse parabolic function that depends mainly in the perceived pH of each strain. This comes from experiments. We know that this shape is what bacteria follow when growing in different pHs. This perceived pH that each strain has, this local pH, is nothing else than the pH in the tank plus or minus a small correction about what they are producing, the acid that they are producing, or that they are producing, or the base that they are producing. Here, this omega is cell sensitivity to this production. And this, if you want, if we come back to the analogy, like in the Daisyworld model, black daisies could live slightly better in a cold planet. Here, acid producers can live slightly better in basic tanks, and the opposite for the base producers, right? And finally, the pH in the tank, the pH free, is nothing else than the income that comes from outside, the base or acid that you put inside the tank, plus and minus the total production of acid and base from the populations that are living in this tank. The results of this model are very interesting. And here, if you see to this surface, you can see in the z-axis the pH free in the tank, and in the x-axis you can see the pH that is the pH income, the pH that enters in the tank. And in the y-axis you can see this gamma is the production of acid or base of this species. When this production is zero, the pH in the tank is non-controlled. So the pH free in the tank matches exactly to the income. But when this production increases, this green plateau appears, meaning that the pH is controlled just at the right amount, no? Just at the pH optimal for this species to exist, mimicking the same the same mechanics as the Daisyworld model. In order to prove, or in order to check the robustness of this model, we added a parasite. We had a third species that would be a completely slave of the environment. This parasite corresponds to this third equation. And if you look at beta, it's not depending anymore on the local pH that the other species can modulate a bit by producing acid or base. This species cannot produce anything, and it's dependent only on the pH in the tank, right? It's a parasite, a slave of the system. The results of that are these two surfaces. If you look at the first one, you can see again pH free against pH income, but now with the corrected alpha, which is the growth rate of this parasite. Even though it's doubled, this growth rate doubles the growth rate of the other species, the homeostatic plateau is maintained, right? Fluctuates a bit, but is maintained. And the second surface, this parasite has different optimal pHs. So it can be an acidophile, an alkalinophile, or a neutrophil. In any case, the homeostatic plateau is achieved anyway. So the parasite don't destroy the homeostatic effect. Finally, a logic jump in the modeling of this of this experiment. We decided to go for the multi-species approach, to increase into the multi-species dynamics. Because we know that the biosphere is filled with species with different pH preferences and different average production of acid or basic compounds. And the pH homeostasis is still observed. So we modified a bit the model. I will not go into detail, but we just allowed the multi-species dynamics to exist. And only you need to think about two mechanisms that were added to this, two properties that each species have here. Each species can have a random contribution to the pH, so they can produce acid or base, not necessarily on their own benefit. And finally, they have different optimals. So in this mixture, we can have acidophiles, neutrophiles, and alkalinophiles. The results of that are interesting too, and the homeostatic effect is also maintained. Here, for example, if you think about the tank where you have this community inside, you have an income of substrate and outflow, but here we don't have an external income of acid or base. We just leave the community inside the tank to readjust, and let's see if they can reach a stable point where they control the environment and the environment allows them to live. So here in this red square, we have the first result, and you can see that in the x-axis, we plotted the community size, and we did this simulation like many times, and we plotted the final events in the y-axis, and different regions appear. For example, when the community size is very small, a huge area of collapse appears, this gray area. This means that the species are not able to readjust and to modify the environment in their own benefit. But when the community size increases, we find this huge white area, which are fixed points, stable points, and the community can stabilize itself into a region so they can control the pH of the tank and allow themselves to live. Finally, in the middle, we have this black region, which we found oscillation and chaos. Just to give you two examples of it, of what are the final, how it looks like the final state of one of these simulations. Here we can see what they look like when they are in these fixed points. You can see here in the first graph in colors, the population fluctuating until they reach a stable point. And in the third graph, we can see how the pH free in the tank is controlled until a value, until a very specific value. The other two graphs are the reconstructed attractors. The second example corresponds to this oscillatory and chaotic region, and you can see how the species oscillate. When you do the reconstructed attractor, you can see that it's chaos. And the pH, even though it oscillates in a wider range, it's also controlled, allowing a population to exist. And finally, if you allow to this tank like an external fluctuation of pH, you add acid or base from the outside. In this graph, this is the blue line. The homeostatic effect is still maintained. So in the first two graphs, we are in this chaotic oscillatory regime. So even though we have this income, this blue line like making noise, the pH is still controlled in a fluctuating environment, but still in control. And these two second examples correspond to these stable points that here are not stable anymore. They're fluctuating because of the external income of acid or base, but basically controlled. So the pH is within a range, allowing a population to exist. And this was all for the theoretical framework of this experiment. And there's only one thing left, is to do the experiment. We are lucky in my lab because we have a wet lab, and we are just starting to do these experiments. I'm happy to announce that yesterday we received like funding to carry it on, and also a PhD to work on it. So very cool. And yeah, I want to do a final comment that now we have this perspective in bioRxiv about synthetic ecosystems, where we discuss, among other stuff, that this environmental regulation, this motif that I should show you along this presentation, can be added in the set of the ecological interactions that we know as competition, cooperation, parasitism, and so on. And this has been all. I want to thank my colleagues from the Complex System Lab members. This is their work too, and thank you all for hearing me, and I'm open for questions. Thank you.